So the first one is, what is the one thing about your field do you think everybody should know?
The one thing about parasite diversity and parasitology, I think it's just how diverse parasites are. There are more species of parasites in the world than there are of free living organisms, because it's the most common mode of life. Because if you look at one species of mammal, it has 2 or 3 host-specific parasites that only occur in that mammal. You can do the same thing with birds that have really specific parasites of birds, or parasites of different kinds of plants. So really, some people have done work and they've shown that there are more, in certain areas, there are more parasite species than there are of host species or free living animal species when you actually do the summary. So I think that's the one takeaway, is that parasites are way more common than we ever expected.
Apologies if you said this in your talk, but what is the maximum number of parasites in like one host that's been discovered?
Oh, the maximum number of species or the maximum individuals?
Well, that's a good question.
Yeah, because we can have literally a person infected with thousands of pinworms, for instance, or Ascaris, the large intestinal roundworm. But probably the parasites that are most diverse as far as number of species goes in a mammal or a bird, we find some rodents infected with 5 or 6 different tapeworms and 3 or 4 different species of protistans, protozoans at the same time. So we can even see more than that. But just from what I know and the things that I've done myself, probably 6 or 7 tapeworms, 2 or 3 nematodes, and 3 or 4 different coccidia in one host.
Let me add to that. We just recently published a paper, so it's also dependent on how much research is done on that specific host. So we know many parasites from humans because of the interest, but we published on the North American deer mouse. There are at least over 100 different species reported, because lots of researchers are working on the North American deer mouse. But some of these South American and not that well-known mammals, yeah, there is only maybe 1 or 2 or 0 reported parasites, but that's not the case. It's just so poorly understood what they have that just there is not enough data.
Right. So there are actually 3 answers to that question. One is the number of individual species in the whole host population, which Gabor answered, or the number of individual parasites in one individual animal, which I tried to answer, or the number of individual nematodes in one animal. So sometimes you can have literally thousands of nematodes in one animal. So it's a 3-part question, which is pretty interesting.
Thank you. We usually have some young people watching these talks who are thinking, I want to do something sciencey, but I don't know what. What would you say to a young person who wanted to follow in your footsteps or get into parasitology? I try and say it.
Yes, if someone wants to work in parasitology, do well in undergraduate school, and then when you go to graduate school somewhere, go somewhere where they have a museum. So go somewhere where you can actually look at specimens that have been collected through space and time. A lot of the work that I've done, I've used specimens that were collected and described in 1820. And so interesting to see these things that people first started collecting and describing and understanding in the first part of the 1800s. And so we can actually get at those specimens because they're in museums. The Berlin Museum, the Vienna Museum, the museum in Geneva, all those places have just deep, deep collections that people have been working over thousands and thousands of hours to put the specimens together. So I would say go to a place, get a good undergraduate education, then go do your graduate degree at a place where there's a museum.
I like it.
Yeah, I like that too. It's fun.
Sue has asked if you are able to study Echinococcus, you can tell I don't know how to say these
Yes.
things, in North America sometime. If you have any preliminary insights, what would you expect to find?
Okay. Okay, ask the first part again.
If you're able to study Echinococcus in North
Yes.
America sometime.
Okay. Well, we have studied a little bit, but what we expect to find is it's going to be in a lot more places than we know now because there really hasn't been a significant survey, a consistent survey over all of the states to know really where it is. So I think we're going to find that it occurs way more places than we expect because we just published that record from New Mexico and it had never been known from there before at all. It's 1,500 kilometers from the nearest southern extent that we knew about. So I think we're going to see a lot more diversity, a lot larger range if we actually get the work done.
And when we think of the future, what do you see is going to happen, or what do you hope is going to happen in the next 50 odd years or so, like in the research and the studying?
Well, both Gabor and I agree on this. We feel that there needs to be a lot more emphasis put into long-term monitoring of ecological and biological processes going on in the field. So Gabor and I both worked on what's called a long-term ecological research project in New Mexico, where we had a big array of areas where we were studying the host parasite diversity over time. And we think that that should be done more intensely over larger spaces on the earth and train people how to do those studies too over time. So I think it's really important to understand what's here, to do surveys and understand what's here before it's gone. I think we're losing it so fast now that we're losing species as we're sitting here talking about it. Gabor, do you have any other comments on that?
Yeah, there are lots of. I might look back that why parasites are important, and you mentioned there that they are the most diverse and most numerous form of life forms on Earth. But it's also it's important to realize that they are a very important part of the ecosystem. So they can have most people think about it as something harmful, something damaging. But in ecosystem, they play a very important role. Sometimes they are stabilizing ecosystems, and sometimes they are actually acting as increasing the host diversity in the area. So you can think of invasive species that they are escaping both their predators and their parasites. They are taking over other ecosystems, and it's an important parasitology research using parasites as a biological control. So it's a very exciting field. But first you have to find those parasites that are very specific to a certain host to use them.
Wow, excellent answers. Thank you very much. I believe we haven't got any more through the chat, so that just leaves me to say, first of all, thank you to the audience for coming and watching tonight's live stream. Thank you for joining us and for your questions. Don't forget that you can buy copies of Parasites, The Inside Story, available from, I'm going to say, Amazon and all good bookstores. The voice in my head says there's one last question. Aha, excellent. We have one last question, which I can do. Leslie said, do you do much work on the effects of the parasites on their hosts?
Gabor and I don't do that kind of work here very much because most of our focus is to do species diversity description and basically species discovery. So our objective is to find out what's there before it's gone. And we really don't do all that much work on how the parasites are affecting the host, but we do have data on that. For instance, I've only seen personally probably 4 or 5 times on all the animals that I've collected and euthanized to put into museums so they can be used by our kids as kids. That's the idea. That's what we're doing, is putting these specimens in the collection so they can be used forever. I've only seen about 5 or 6 times where the parasite was actually causing the host enough distress that it actually made the animal so I could catch it by hand without even trying. I was able to just walk up to it and pick it up. Of course, mange, which we didn't even talk about, the mites. Mange causes really big problems in domestic animals and in pets and wild animals. And so that really does cause some big problems. But those are mites, and we don't generally work on mites too much, although I've described several mites with some of my colleagues. But our most of our descriptive work is mostly done on trying to determine what's there and get it described before it disappears. So I'm sorry I can't tell you too much about how parasites might affect the hosts. What I do know is that most of them that we see do not affect them very much at all. Really low impact of the parasites in the host. However, the definition of a parasite is one animal that lives inside or on another animal that causes some harm. But the harm can be minuscule, like 1 ATP out of every 300 for energy use, or it could be massive. It could be using all the energy of the animal up because it has a huge infection. So it could be almost nothing, or it could be really a lot. So parasitism is hugely diverse. It could be almost nothing to massively life-threatening. Like malaria. Malaria kills, it infects millions of people per year, and falciparum malaria kills 200,000 kids per year in the world. And that is a protistan protozoan parasite that's transmitted by mosquitoes.
We've actually had some more questions come through, so I was jumping the gun when I was logging off, so apologies, everybody. So Petra has asked, is there much investigation being done at sewage treatment works to identify human parasite?
There has been a lot. I'm not sure if it's ongoing right now, but there's a lot had been done in the past. What has usually been done is they put the material through the sewage treatment plants to then deactivate any of the parasites in there before then they take the sludge out and put it on fields. So there is some work being done there. And so they're trying to determine what's in there before they do the treatment so they can make sure that they deactivate all those parasites that are coming through. Of course, the things that are coming through can be everything that we have. Like I had Ascaris one time, nematode about that long. And I didn't know I had it until I did a quick fecal check, and there was some eggs. And I thought, oh my God, that's from me. So I went to the doctor, and the doctor said, oh, we don't have that here in North America. So that's not what you've got. And I said, well, I have a slide. Would you like to see it? So he did, and I got my treatment. But he wasn't going to believe me at first. It was very interesting.
Wow.
He told that to the wrong guy.
Brings out the research paper.
Yes, here's the paper.
Becca has asked, are there loads of parasites you've not discovered yet?
Oh, yes, probably 90%.
Wow.
Yeah, so we really don't know very much. If we went in any of these places that we go, have new parasites. For instance, just in Nebraska, 400 kilometers to our west, I collected small mammals out there, and they had new parasites in them, every single one. So it's just so much is unknown that it's really mind-boggling.
Gabor, were you going to say something?
Yeah, actually, I wanted to emphasize, so we are museum people, and this is a reference collection for parasites. So that's our most valuable and most available resource for research, that we can look at other parasites in the collection. And we have lots of visitors who are using our collection or this collection to compare whatever they are finding. And every year, because when people are finding something new, they have to deposit it in a museum. Every year we are receiving new material, new species that they are describing. And as Scott mentioned, that it's it's surprisingly a lot of new things are described, sometimes even in well-researched areas in North America or in Europe. It continues, so it's there are still lots of species to discover. So yes, if someone is interested in this kind of research, there are opportunities to discover new species.
Roughly how long does it take between someone discovering what they think is a new species and it coming to the museum and being like confirmed?
Well, Scott is publishing a lot. Behind me, there are some drawings. So it can be done within, if you have the specimen, in 3, 5, 6 months. Is that right? The publication process, Scott?
Yeah, it can be done very quickly. However, it can be longer too, because I collected some specimens in Bolivia in 1984, and I thought I would get more individuals of the tapeworms that I got from those cute little tucos, and I never collected them again. And so I waited until just this year to publish the paper, because I thought I would get more, but it turned out I wasn't going to. So I published the paper now, so it was like 40 years later.
Wow.
Well, maybe not quite that long, 30 years.
Leslie has asked, so are some parasites beneficial?
That's a good one. Some parasites, usually, well, the definition of parasitism is something that causes harm. So it might be a symbiont then, if it's living with another animal, so we wouldn't necessarily call it a parasite. However, some parasites do act as population regulation in host populations. And so in that case, it could be called beneficial in the ecological sense. But if we use the fairly strict definition of a parasite, it's got to cause some harm. However, there are some nematodes that live in rodents that only eat material that's in the cecum, in the large intestine of the rodent. And they digest some of that material and then excrete. They use 2 ATPs out of every 38 that are in a glucose molecule. They use the 2 ATPs and they excrete the rest as lactate. And lactate is then absorbed by the intestine of the rodent. Therefore, what would normally have just passed out in the feces gets actually given back to the rodent. So it could actually be using up some of the ATPs of the rodent, but it actually may be giving more back than it was actually taking. So that would be a really good area of research for someone. It's not my area, but it's kind of a logical summary of what happens with those things. Those are called pinworms, in fact.
Yeah. I think another analogy, the enemy of my enemy is my friend. And I mentioned biological control, and they are using certain nematodes to control grubs in the soil or insects in the soil.
Yeah, that's a good one. Steinernema is the genus, and they grow those up in huge stainless steel vats, then dry them down, and then you can buy them. You can go online and buy Steinernema, and you can then spray it on your yard, and those little things come alive, and then they act as like little smart bombs, and they go into the insect, release bacteria in the insect, then kill the insect, then eat it from the inside out.
Wow.
Well, they don't eat the insect. The bacteria kill the insect. They eat the bacteria.
Well, there you
Very
go.
cool. Yeah, one of my favorites.
Rupert has asked, the little fella that you dissected or showed in Mongolia, do you have any idea how much longer it would have survived and how long it would have been infected for?
Oh, that little guy. He was pretty sick. That was an example of a vole that didn't look so hot. When I picked it up, I could feel that he wasn't really in very good shape, but he probably would have lasted long enough to get eaten by the wolf that was living in the area. So once they get that big of an infection, they probably can't run as fast, and then they become easy prey for some other animal. Probably if nothing would have eaten him and he would have stayed in a nice little spot, he might have lasted another 3 months, but the liver was almost gone. So pretty interesting. But now he's in a museum forever.
Yeah. Voles have really short lifespan, 3 to 6 months. as an average.
Mm.
And there are also it's interesting because there are also studies that they were looking at parasite infected animals are more likely to be predators are more likely to catch them. So there are even some ideas that these parasites they affect the host in such a way that they are easier to catch. And also, it's if you are comparing predation on the host, parasite infected individuals are more likely to be caught. So, and that's actually what happens, that uninfected individuals are more likely to survive. So the healthier individuals are more likely to survive.
We talk about that in our book too. There's a chapter in there about how some of these parasites can influence the behavior of the host to make sure they get onto the next host. So it's pretty interesting there. So, and there are some parasites that live in snails that eat other parasites that come into the snail. So there's a biological warfare going on in the snail itself. So these things called echinostomes will attack other parasites coming in as they're trying to make their home inside the snail itself, but the snail kills them and eats them.
Wow.
I'm sorry, the parasite inside the snail kills and eats the other trematode coming in. So we know that, but it's not it's really not possible to understand everything that's out there.
Absolutely.
Yeah. So
LJ has asked, with the larger parasites, it may be easier to identify an infection, such as the picture of the liver that was mostly cysts. How difficult is it to find protist parasites that, as they are quite small, do you look for other signs of disease or telltale behavior in hosts?
Well, yes. Some, like the protistan parasites, the way we find those in the feces of an animal, we don't know that it's infected when we look at it. So what we have to do is take a little fecal pellet, and we then put it into a centrifuge, and we have to spin it down with sugar solution, and all the parasites float to the top of the centrifuge tube, and then we have a cover slip on it, and we take the cover slip on it and put it on a glass slide, and then we put that glass slide on a microscope, and then we examine it to see if we can find the parasites. So that's one way. Another way is if it's a blood parasite, we take a blood smear. We take a very small bit of blood from an animal, a live animal or a dead animal, and then we put it on the slide, and we let it dry, and then we bring it back to the lab, stain it in Giersche solution, and then we examine it with a microscope to see what kind of parasites, what kind of blood parasites it has. We do the same thing if it has ectoparasites. When we put the animal, we can look at it fairly carefully. If we are able to brush the animal before if it's not going to be euthanized, we can find what kind of ectoparasites live on it: lice, fleas, ticks, mites, all those kinds of things. So there's lots of different ways that we don't know they have something until we actually look at it really much more carefully using different methods. Good question.
Makes your job a lot harder, I assume.
Oh, it's really not easy. I mean, in order to do this, we have to know a lot. I mean, it's really mind-boggling. But it's so much fun because everything's different every day.
Excellent. Luz has asked, do you always look into insect parasites such as cordyceps?
Well, I have only looked at them because of interest. In Bolivia and South America, I've found ants that have the big old fungus growing out of them, and so I thought, well, that's cool. I take a picture and I can show it in my my parasitology class when I'm lecturing, but I don't investigate those myself. Gabor, do you have any experience? I guess we don't really look at
yeah, insects are very important. It's a very big taxonomic group, and you have to be more specific because some of the tapeworms, the intermediate host is insects. So we have a beetle colony that we are keeping around in case we want to. So these insects might feed on the feces. They pick up the tapeworm egg, and the larval tapeworms start to develop inside the insect. And it's really, when you dissect them, you can see these under the microscope, you can see these tiny, tiny larval tapeworms wiggling out from the insect. So that we did that, but they also, you can see that there are protozoans in insects, bacterial parasites, all the way. Yes, and even nematodes can live. So we have behind me, there is a collection of nematodes from bark beetles, and they were exploring that for controlling bark beetle infestation. The same idea, using nematodes as a natural enemy of bark beetles.
Yeah, that's pretty cool. And the insects, the beetles of the world many of them are infected with these little things called gregorins that are related to coccidia, which is related to malaria. So these gregorins occur in many, many beetles, and probably the fauna is just beginning to be scratched, the surface of the number of species that exist.
I have one more question that we can fit in. And it's from Valeria has asked, how do you think we should integrate the biological invasions or rapid geographical range expansion in the Dharma protocol?
Well, that's a good question. I guess what she's asking is, how do we deal with rapid range expansions, or how do we detect them? There's a couple of different ways. One of them is if we're seeing things jump up where we don't expect it, where we're seeing species. That's if we're using the Dharma protocol, we have a documentation and assessment and then monitoring. If we're monitoring and we see something come up in an area where we don't expect it, that would be an indication that we can then take action. And then we can look at other places that it might have. If we're actually implementing the Dharma protocol over broad geographic space, we could then say, let's look at this similar geographic or similar ecological area in this part of the world, in Europe, for instance, in the Alps. If the same thing is happening there that's happening in the Rocky Mountains, we can then know something big is going on. So geographic expansion is an interesting concept, and using the Dharma protocol, the monitoring part would be able to detect those really quickly. So I think if we implement Dharma over large geographic spaces, it would be really exciting.
Excellent. Well, thank you for coming and watching. If you do want to watch any more of our live streams, you can go to our What's On page. It's just rigb.org. So thank you, everybody. And of course, thank you, Scott and Gabor, for your talk tonight as well. That's it from us. And I'll say goodbye.
Thank you very much. And
Thank you very much.
Contact us anytime at email and we'll respond. Thank you.
Thanks. Bye.